Hello and welcome everyone to my YouTube channel. Today's lecture will be on cell cycle and its checkpoints. Let's proceed. The eukaryotic cell cycle. The cell cycle refers to the cyclic occurrence of a series of events leading to cell division. The cycle is required for normal growth, development and maintenance. The cells which divide continuously are the intestinal cells and the skin cells. These are the two examples. The cells that divide under specified conditions are the lymphocytes, and the cells that do not divide at all are the cardiac muscle cells and the nerve cells. So, cell division is dependent on the type of tissue and their functions. Let us look at the cell cycle and its phases. The cell cycle phases last for around 20 to 22 hours. As you know that the cell cycle refers to a cyclic occurrence of series of events leading to cell division. So there are four phases in cell cycle, as you can see here: the G1 phase, S phase, G2 phase and the M phase. These are the four phases of a cell cycle. Of these four phases, all form the interphase except the M phase, which means G1, S and G2 phase are the forming the interphase. During this phase, the cell prepares for cell division. The actual division occurs in the M phase or the mitotic phase, in which both DNA and cytoplasm divide to produce two daughter cells. The activation of each of these phases is dependent on proper completion of the previous phase and progression into the next phase. If there is any error, the cell temporarily stops dividing and it goes into a state of dormancy, which is known as the G0 phase. This is also known as the state of quiescence. During this stage, the cell repairs the damage. However, if the damage is not repairable and it is too large, then the cell undergoes apoptosis. So this is a defensive mechanism which prevents transmission of damage into the next generation. So apoptosis is very necessary and it prevents transmission of damage and mutations into the next generation. So cell cycle is precisely a regulated process, and if there is any disturbance in the regulation, then it will lead to many abnormalities. So we will quickly look at the different events that occur at each phase. In G1 phase, protein and RNA synthesis occurs and it lasts for 6 to 12 hours. In S phase, DNA duplication, protein and RNA synthesis, all the three processes occur and it lasts for 8 to 10 hours. The S phase commences with the start of DNA synthesis and the entire DNA is replicated here only once. Now in the next phase, which is the G2 phase, there is no DNA synthesis because the complete replication has already occurred in the S phase. However, protein and RNA synthesis continue, and also there is arrangement of microtubules that are required for formation of uh, the spindle. So this stage occurs for 3 to 4 hours. The M phase is the shortest phase and it is about 1 hour in duration. And during this phase, the chromatids separate and there is cytoplasmic division. So the cell divides into two daughter cells. This is the complete cell cycle with its four phases. Now we come to checkpoints. The cell cycle is controlled at a series of checkpoints that are the mechanism to monitor the order integrity, and fidelity of the major events occurring in a cell cycle. There are various checkpoints, but the three major ones are the G1S checkpoint, the G2 checkpoint, and the M checkpoint. What are these checkpoints? Just like our traffic is regulated by traffic signals, same way our cell cycle is regulated by various signals at different checkpoints. And there are certain requirements that need to be fulfilled at each phase of the cell cycle, and the cell cannot enter the next phase of the cycle unless these requirements are met. Cell cycle controls or checkpoints. The checkpoints in a cell cycle are very important because they help to uh, halt the cell division process if there is any genetic damage, and it allows the cell some time to repair the damage before cell divides. If the damage cannot be repaired, then the cell undergoes apoptosis. Now, this is the cell cycle showing different checkpoints which we will discuss in detail. So, there are three checkpoints in a cell cycle, and these three checkpoints are the G1 checkpoint, the G2 checkpoint, and the M checkpoint. So, we will first look at the G1 checkpoint. The G1 checkpoint determines whether all conditions are favorable for the cell division to proceed. The G1 checkpoint is also known as the restriction point at which the cell irreversibly commits to cell division. Cells set up certain requirements to be fulfilled by the cell to pass through the checkpoints. There are certain external factors such as growth factors which play vital role in carrying out the um, uh, cell transition from G1 checkpoint to the next phase. The cell will only pass the checkpoint if it has the appropriate cell size and adequate energy. So this checkpoint looks at whether there is sufficient nutrients, growth factors, and if the DNA damage is there or not. It checks for all these three things, and once this is crossed, then it enters the next phase. If the G1 checkpoint is also known as the G1S checkpoint because it's this checkpoint exists towards the end of the G1 phase and in the beginning of the S phase. Now we look at the G2 checkpoint. The G2 checkpoint ensures that all the chromosomes have been accurately replicated and the replicated chromosome is not damaged before the cell enters mitosis. So the things that G2 checkpoint looks for are cell size and DNA replication. If the checkpoint mechanism detects any problem with the DNA, the cell cycle is halted and the cell attempts to complete the DNA replication or repair the damaged DNA. And now we come to the metaphase checkpoint, which is also known as the spindle checkpoint or the M checkpoint. Now, this spindle checkpoint occurs at the end of metaphase. This is towards the end of metaphase. And this M checkpoint determines whether all the sister chromatids are correctly attached to the spindle fiber before the cell enters irreversible anaphase stage. At the end of the stage of metaphase, the spindle fiber arising at the opposite pole of the cell attached to kinetochore in the equatorial plane. The M checkpoint also involves signal from cyclin-dependent kinases. So the regulation of cell cycle is controlled by regulator molecules that either promote the process or stop the process. Cyclins and cyclin-dependent kinases. The cell cycle is regulated by a complex of proteins called cyclins and cyclin-dependent kinases. At least 10 different cyclins are known and 8 different cyclin dependent kinases are known. So these are two classes of proteins that control the progress of cell cycles, and these proteins, as I told you, they are known as cyclins and cyclin dependent kinases. These are synthesized throughout the cell cycle and hence they are named as cyclins. Now we will look at the cyclins and CDKs. The cyclins activate CDKs which phosphorylate specific regulatory proteins. So the work of cyclin is to activate the cyclin dependent kinases, and the role of cyclin dependent kinase is to phosphorylate the regulatory proteins. We know that there are different cyclins present, but in this lecture we will be discussing the four major types of cyclin, that is A, B, D and E. These are the regulatory cyclins, and there are five different cyclin dependent kinases we will discuss, that is CDK 1, 2, 4, 5 and 6, which control the cycle. These are catalytic units, serine, and they are known as serine threonine kinases. So the cyclins and cyclin dependent kinases may occur freely, independent of each other, unbound to each other, or they may be bound to each other. When they are in the unbound condition, that is the cyclin and CDK are separate, then it is an inactive stage, and when they are in the bound state, then they are in the active state. Cyclins and cyclin dependent kinases play a very important role in the regulation of cell cycle. They are a family of proteins whose concentration increases and decreases at specific phase of the cell cycle. This is the cell cycle, and now we will see the roles of different cyclins. So cyclin D activates two kinases, CDK4 and CDK6, at the late G1 phase. Once they complex together, they are active serine threonine protein kinases which activate various substrate. One such example is the retinoblastoma protein, which is activated by this complex. The role of retinoblastoma protein will be discussed later. At the G1S checkpoint, it is the cyclin E that is complexing with CDK2. The cyclin E has a very short lifespan, and it rapidly degrades and releases the CDK2. This CDK2 is now free to bind with the cyclin A, and it regulates the S phase. It is very important for the S phase, and because of this complex, the initiation of DNA synthesis occurs at the S phase. This is the G2 checkpoint of G2 phase, and in this G2 phase it is the cyclin A that activates CDK1. At the M checkpoint it is the cyclin B CDK1, and the cyclin B CDK1 R complex is also known as the rate limiting complex because it is very important for the G2M transition in the eukaryotic cell cycle. Thank you for watching. Like, comment and share, and subscribe to my channel.